I'm a lab head here at the Garvan Institute. I work primarily on breast and prostate cancer, and I don't work on ocular melanoma. But what I do know about is single-cell sequencing, and so Anthony asked me to come along to see whether there are some opportunities to explore using this technology in the area of ocular melanoma. So I'm going to give a kind of a high-level overview of the technology, how we're using it in some disease settings, and then try to give some ideas about how we might apply it in the melanoma space. So, I think we have a good appreciation now that tumours are not just homogeneous masses of malignant cells, but they're in fact essentially ecosystems of diverse cell types, both the well-established cell types that we know, fibroblasts, endothelial cells, and so on, but we also see significant heterogeneity, both genomic and functional, within the malignant populations of tumours. And so by using single-cell methods, we can start to distill our studies down to what is essentially the quantum of life and disease, which is the cell. And by doing this, we think we're going to have some opportunities to gain new insights at cellular resolution. So, for example, through the identification of rare malignant populations that in bulk studies might be lost. We can define novel cell types, novel states that cells, cells can occupy. We can start to map cell-cell signaling events once we've got single-cell data. And importantly, we can start to understand context. So, for example, we can ask, within the proliferating population of cells within the cancer, what are the features of those cells, what's making them proliferate, what therapeutic targets might they express, and so on. And we think that single-cell is going to make a really big impact on a number of facets of clinical management of disease, both in terms of prognosis, understanding drug resistance and response, and ultimately in identifying new therapeutic strategies for patients who don't respond to conventional care. So I like to use a political analogy to try to hammer home why we think single-cell analysis is of value. So if you think of the United States political system, we think of Texas as a red state, always has been, always will be. But the problem is that model doesn't really explain the kind of cultural and political and economic diversity that we observe within Texas. And so once you start to break your analysis down to higher resolution, so in this case down to the electorate level, we start to see, for example, a belt of Democratic electorates along the Mexican border, and we can identify this little island of blue within a sea of red as being Austin, which of course is the home of much of the kind of arts and culture within Texas. And so we think this is an analogy that in essence what we're trying to do is cellular epidemiology. Can we go at higher resolution to understand the bad cells, the important cells, and their interactions within a tumor? So this area has moved very quickly, and now there are modalities and technologies to study essentially any feature of cancer biology that you might be interested in, be it gene expression, genomics, proteomics, and so on, at cellular resolution. And I think because of this, it's become a really interesting nexus between disciplines. So this is not simply genomics. It's not simply cell biology. And in fact, to be honest, the way we do single-cell sequencing, we view this more as an extension of pathology. That's the kind of lens through which we view what we do. And so it's a really interesting opportunity to bring together people with varied expertise and for them to gain insights in their area from these kind of data sets. So we're very lucky here at Garvan to have a really world-class center in which to do single-cell sequencing, and this is the Garvan Weissman Center on level 11 of the Kinghorn just next door. It's a joint initiative of the Garvan Institute and the Weissman Institute of Israel. This is a $10 million facility, and in fact it's just been named as one of UNSW's future institutes as well. So lots of resources going into this area, and this now gives us access to expertise and technology, but importantly also the capacity to do studies at scale. So we can really do cohort size single-cell sequencing studies now with the opening of this facility. Okay, so I thought I'd briefly show you one of the ways that you can do single-cell sequencing studies. This is the method that my lab uses predominantly. And this little kind of DVD player at the top here is actually the busiest machine up on level 11 of Kinghorn. It's the Chromium Controller from 10x Genomics. And essentially what this does is it lets us do molecular biology reactions in massive parallel. And so instead of using a tube for per sample, what we do is we use a technology borrowed from digital droplet PCR, actually, where our reaction vessels now become nanoliter droplets, aqueous droplets containing a cell and a capture bead within an oil emulsion. Now, I don't want to go too much into the details here, but essentially what that means is that we can now take thousands of cells from a specimen and in parallel do single-cell molecular biology reactions, so for example, to make transcriptome libraries that can then be pooled, sequenced en masse, and then the data from that can be taken and reallocated back to the individual cells from which it came. So this can be very efficient, very rapid, and generate seriously big data for us. So the core of what we do in my lab is single-cell RNA sequencing, and so this allows us, for example, to generate whole transcriptomes, so entire gene expression profiles from thousands of genes from a single specimen, and that's important. It's an unbiased assessment of the cell state and its expression of disease-relevant markers. But of course, there are limitations here as well. RNA doesn't tell us about protein, there are sensitivity issues and so on. So because we're working with clinical specimens, we want to try to bolt on additional modalities to learn as much as we can from that specimen. So the first one is to get some proteomic data in parallel to the RNA data. So here we're using a really cool new method called CytSeq. And essentially this is similar to mass cytometry or flow cytometry, but instead of attaching a heavy metal or a fluorophore or a chromophore for that matter, like in IHC, to an antibody, we attach a unique DNA barcode that's polyadenylated. So essentially when the antibody binds to the cell, cells get captured, and these barcodes from the antibodies go into our sequencing library and get sequenced. And so within that library, the frequencies of these barcodes tells us how many antibodies bound to that cell, which is proportional to how much of the antigen was on that cell. And so this is a really kind of stunning technology now to give us parallel RNA and protein measures. My lab, together with Chris Goodnow and Martin Smith here, have also developed a method to do targeted full-length sequencing from single cells. We call this RAGE-seq, and this can be important when the information contained in these libraries isn't enough to tell you what you want to know. And probably relevant to uveal melanoma, the kind of things you can study, for example, are splicing or somatic mutations of genes of interest. So you might want to make an SF3B1 capture probe to pull that molecule out and to sequence that in specifically very high detail, and perhaps even some of its targets, because it's a splicing regulator, to see whether they're altered in your disease. And you can tailor that to your needs. And finally, the field is, of course, coming back to tissue, because we need to understand things in spatial context. And there are some really amazing new technologies emerging now to not just validate findings back in tissue, but actually use tissue as a discovery platform itself. So here's an example of some data that we've generated together with our friends at the MIA. This is metastatic cutaneous melanoma, but I think it demonstrates just some of the points that I'm talking about here. So this is known as a UMAP depiction of the data. As you can imagine, every dot here is a cell. So we have thousands of cells from this sample. Every one of those cells we've measured more than 20,000 genes, so this is a very big data matrix, and this is a way to start to visualize it. So cells that have similar gene expression features cluster together, and so you can see quite simply that now we can identify myeloid cells and fibroblasts, B cells, T cells. But interestingly, within the malignant population here, there's actually quite remarkable diversity. So perhaps as pathologists would have told us by looking down the microscope, morphologically we can see diversity, but we can now see that in terms of their actual gene expression features. And so now we can start to do simple things like say, well, tell me the gene expression features that explain or drive this diversity. And so this is almost like digital pathology, where we can just dial up whatever gene we want that you might be interested in. And here we're projecting in red the expression of just markers we've picked off the top of our head back onto this space here. And so you can see straight away, for example, that these are the proliferating cells here, Ki67 positive, aurora kinase positive. So that's the ones over here. And then we've got, markers of differentiation, MITF and tyrosinase and so on. And they're segregating into different populations. So we can start to functionally annotate these different populations and start to try to understand which of these we think are the bad players, and so the ones that we really want to zoom in on and understand more about. We can also infer copy number change using this data. This is an example from breast cancer, but it could very easily be a melanoma. You can see again in the breast cancer data, we're seeing heterogeneity within the epithelial population here. So we're trying to figure out which of these are contaminating benign cells, which of these are malignant. And so on the right, you can see we've inferred copy number change, gains in red, losses in blue. These are the clusters lined up on the side here, and these are chromosomes along the x-axis. And so straight away you can make a few observations. The first is which the cluster 6 is almost contaminating, certainly contaminating benign cells. There are very few copy number changes there. We can see in comparison cluster 7 has very dense predicted copy number change, so probably a very unstable, genomically unstable population of cells. And then interestingly, if we look up in cluster 0 here, that I'm highlighting in the top right, we actually see evidence for subclonal diversification in copy numbers. So here this loss is only found in half the cells found in that cluster. So immediately starts to help us tease out the drivers of heterogeneity within the malignant cells. Okay, so finally I'll just return to this CytSeq method and its application to tumor immunology, which of course is maybe not a huge area of research in uveal melanoma, but I'm sure it will have specific applications. And we're using this in a number of different settings. And so this is this CytSeq method where we can measure protein abundance in parallel with transcriptomes. One of the reasons we do this, of course, is that immunologists just love facts, and if you can't show them something that looks like facts, they won't believe it. And so the beauty of this is that the data we generate with this method links back to those decades of that canon of knowledge in immunology that we can link through the data we're getting from single cell. It's very sensitive, and what's really amazing is there's no theoretical limit to how many antigens you can go after. You don't have the problems of fluorescence overlap, for example, that you have with flow cytometry. And so in fact, we're using a panel of 130 antibodies now, so we can really go after a lot of different antigens at once and measure them simultaneously. And of course, that's important if you're thinking in oncology. You want to know that the therapeutic target that you're interested in is actually present at the protein level on the cells that you think you want to drug. And so to show you the kind of benefit that we gain from this, I've just pulled out the lymphocytes from a triple-negative breast cancer here. This is what RNA data will show you. We can discriminate B cells and T cells. That's great, but there are limitations to what we can learn based on this. Whereas if we now layer in the protein data, you can see that we have a much greater capacity to reveal fine granularity within these cell types. And I'll draw your attention, for example, to the CD8 T cells up here. In fact, we can now discriminate two different populations of what look like tissue-resident T cells based on CD103 expression, a PD-1 low and a PD-1 high. And so I think this is actually a novel observation that was actually facile to make, and I think it reveals the opportunities that this kind of technology can provide in discovering new cell types that might be relevant to immunotherapy response in this case. And this is just kind of mapping, again, any one of those 130 antigens that you're interested in back into these different populations. You can measure them, and so here, for example, is that PD-1 data that I'm pointing to above. Okay, so one of the important considerations is that single-cell sequencing methods really are very sensitive to the way you've stored tissues. Snap-frozen tissue can be studied using some of these modalities, but it's not easy. Paraffin-embedded tissue, it's very difficult. There are very few options to use single-cell methods, and so I think it's important for people who are interested in future-proofing their biobanking to get on board to really think about how they do that. And so we've done an experiment with our colleagues at the MIA. They've got a very active and very sophisticated tissue banking program operational and been running it for a number of years. And we wanted to check that the way they were banking tissues is compatible with single-cell sequencing. And so we've done some experiments where we take a freshly resected melanoma, we process it immediately and capture it. We store it in the fridge overnight and then process it and freeze it the next day. And finally, we cryopreserve it immediately. And I won't show you the data in the interest of time, but essentially what it shows us is that all three methods give us comparable data quality, which is great news because it means many of the retrospective samples that they have are now amenable to the kind of methods I've shown you. And so using these technologies now, we have a number of active clinical single-cell programs in my lab. We've got a large program in breast cancer, essentially sequencing 1 million cells from 200 surgically resected breast cancers done here at St Vincent's and at Chris O'Brien Lifehouse. We're also starting to look in metastatic disease, both through biopsies as well as through a rapid autopsy program, a national program that we run. But I think most interestingly is starting to use single-cell technology in the context of treatment to understand treatment resistance and gain really new mechanistic insights. And we've got a number of programs there. We're sampling breast cancers during either neoadjuvant chemotherapy for ER-negative cases or endocrine therapy for ER-positive cases with LG and Lim. With Anthony, we've kicked off a couple of prostate cancer projects, again, for early disease as well as longitudinal sampling of metastatic disease during a clinical trial, which I think is going to be really exciting. We're also doing a project with Georgina and Richard over at the MIA, looking at longitudinal sampling of patients receiving adjuvant immunotherapy to understand why some respond to immunotherapy and some don't, so to try to get better predictors of immunotherapy response. And we've also collaborating with Joseph Powell's group on a similar project in lung cancer. So just to summarize, I think I hope to have kind of convinced you that there are many methods now to study the kind of area of biology that you might think is important at cellular resolution, and that many of these can be multiplexed to get maximum information from a single biopsy, for example, that you might have access to. And that this can give us really new insights into the heterogeneity of the tumor microenvironment and of the malignant populations. And I'm absolutely confident that this technology is and will revolutionize translational cancer research. And so I hope we've got some opportunities to apply it to your research. But we need to think about the way we're banking tissues to make it suitable for downstream analysis. So I'll just finish up by acknowledging the wonderful people in my lab, as well as collaborators, including Georgina, Richard, and Inesh from the MIA.